Buenos días a todas y todos. Good morning, Professor Joseph, and thank you for joining us today. Muchas gracias de parte de ILSI Mesoamérica y en especial del Comité de Inocuidad de los Alimentos. Le damos la bienvenida a todos ustedes quienes nos acompañan en este penúltimo seminario del año y agradecemos por supuesto al profesor Joseph Baumert, quien será nuestro conferencista el día de hoy, y al profesor Mauricio Redondo, quien nos colaborará siendo hoy el coordinador de esta sesión y en especial en la sesión de preguntas y respuestas. Siempre hacemos una recopilación de lo que es ILSI para recordarles lo que somos. Somos una organización sin fines de lucro que nos dedicamos de manera especial en temas que tienen que ver con la ciencia para proveerla y para generarla y comunicarla, como es el día de hoy. Hoy estamos haciendo una actividad de crecimiento en capacidades técnico-científicas que esperamos tengan un impacto positivo y mejoren la salud pública, el bienestar y por supuesto que apoyen también el desarrollo sostenible en todos nuestros países. Si quieren saber más de ILSI, pueden ir a nuestra página web ilsimesoamerica.org y por supuesto también a nuestras redes sociales. Siempre también hacemos una declaración de exención y es que las opiniones expresadas por las personas, en el caso de hoy el profesor Joseph, lo hace en su nombre personal y no lo hace en nombre de ILSI Mesoamérica, y también pues él actúa de manera voluntaria. Él pertenece a la Universidad de Nebraska, ahorita el profesor Mauricio lo va a presentar, y bueno, ha tenido total libertad, es responsable de su presentación y no ha recibido ninguna remuneración por estar el día de hoy con nosotros. Así que le voy a pedir al profesor Mauricio que presente al doctor Joseph. And again, thank you very much, Professor Joseph, for joining us. And well, Professor Mauricio will be presenting you. Thank you.
Muchas gracias, Hania. Bueno, es un placer para mí presentar al doctor Joel Baumert de la Universidad de Nebraska. El doctor Baumert es actualmente profesor en el Departamento de Ciencia y Tecnología de Alimentos de la Universidad de Nebraska-Lincoln en los Estados Unidos, y también es el director del Programa de Investigación y Recursos sobre Alergias Alimentarias, conocido por sus siglas como el FARP. Él tiene mucha experiencia en el tema de alérgenos alimentarios, ha mantenido programas de investigación y extensión, tanto a nivel nacional como internacional, activos durante los últimos 15 años, y su investigación se ha centrado más que todo en el desarrollo y mejora de métodos analíticos para la detección de proteínas alimentarias alergénicas y el desarrollo de modelos cuantitativos de riesgo, que es el tema principal de esta charla. Las actividades de divulgación del Dr. Baumert incluyen capacitación para la industria alimentaria sobre soluciones de control de alergenos destinados a mitigar el riesgo de contacto cruzado de alergenos y consultoría de evaluación de riesgos cuantitativos. Yo conozco al Dr. Baumert desde hace varios años, durante mi oportunidad de realizar estudios de posgrado en la Universidad de Nebraska, y desde ese momento yo conozco su gran expertise en el tema de alergenos, por lo cual es un lujo tenerlo hoy aquí con nosotros. Joe, I'm going to proceed with the introduction in English. Dr. Joe Baumert, he is a professor at the Food Science and Technology Department in the University of Nebraska-Lincoln in the United States. He is also the director of the Food Allergy Research and Resource Program, also known as FARP, in the same university. He has coordinated research and extension programs both nationally and internationally during the last 15 years as a faculty member at UNL, and his research has centered mostly on the development and improvement of analytical methods for the detection of allergenic protein in food and the development of risk assessment models also for food allergens. His extension activities include training for the food industry in terms of control or solution for control of allergens, aim to mitigate the risk of cross-contamination of allergens, and also a consultory of assessment of this risk in foods. I also mentioned that I met you some years ago while I was doing my graduate studies, and for from that time, I had the opportunity to know about your expertise and all the work that your group is doing. So thank you very much for your time and disposition to share your knowledge with us. And now you can start with the webinar. Your microphone, Joe.
There we go. All right. Well, Hania and Mauricio, it's a pleasure. I thank you for your kind introduction. Always a pleasure to connect and provide some insights on food allergens, food allergen research that's going on. I think this is an exciting time in the area of food allergens, as a lot of the research we've worked on over the years is now being utilized, implemented on an international basis. So today I've entitled my talk as Food Allergens, What's New? Really in that space, it's a lot of discussion about risk-based approaches for labeling considerations, such as those that would be for considerations on what's a priority allergen, what is the use of precautionary allergen labeling, or PAL as it's been termed internationally, and how can we consider some of the science that incorporates that risk-based approach. So what does it mean to have a risk-based approach? What I'll be discussing today primarily would be centered around the work of the FAO-WHO expert consultation on risk assessment of food allergens, in which I was an expert committee member that participated through the series of different tasks that we were asked as a function of the Codex Committee on Food Labeling. So it's a pleasure to be here to talk a little bit about that work and some of the aspects on where that research underpins that work as well. So I think as we dive into risk assessment, I think it's always important to ground ourselves on what is it that we're talking about when we think about risk. And certainly we think about that hazard, and I use some nice slides that you'll see coming up here that were provided by EFSA a number of years ago that kind of illustrate what hazard versus risk mean. So hazard, of course, is something that has the potential to harm you versus that risk, where the risk is the likelihood of a hazard causing that harm. So again, what does that really mean in terms of risk assessment? So let's dive a little deeper into that. So I provide this one. I really do enjoy the illustrations that EFSA provided, kind of simplistic and allow us to look at it. But here you see sharks, something that here in the very central U.S., here in Nebraska, we have no sharks, but it's a nice illustration nonetheless for us to think about. So if we think about our scenario here, if I'm standing on that beach, certainly a shark in the sea is a hazard, but is it an actual risk? Since I'm standing on the beach, I don't have that risk of getting bitten by a shark. It's not until I enter the water where that shark may be that there is that risk that it can potentially occur, again, depending upon a number of variables. Another way to look at it, here's another illustration. If we think about lightning and during a thunderstorm, certainly if I'm indoors, I have certainly a hazard that the lightning could strike, but is there a risk to my health in that case if I'm indoors? So if I take that scenario and I stand under a tall tree during that thunderstorm, I enhance my risk of getting struck by lightning during that thunderstorm, because I've put myself into that environment in which there's that opportunity for that exposure and risk. So with both of these, I think the key that pins these together is we think about hazard, but we have to think about that exposure scenario. Are we exposed, and to what degree are we exposed? Those two, hazard plus exposure, come together to think about that probability of risk. So what we'll talk about today is really bringing about the science on how to think and calculate that potential exposure risk. So let's think in terms of allergens. So I'm going back to my scenario on the beach versus being in the water. And a good colleague of mine, Dr. Ben Remington, adapted this from the EFSA slides that you saw earlier. So if we think about allergens, we could say, well, may contain sharks. So just like we think about with allergen cross contact and that potential for residue to be present, we could say may contain peanuts as an example. But there can be more put into the thought process when we go through that evaluation. So you could ask, was the sign placed after a qualitative or maybe a quantitative assessment? So qualitatively, sharks may be in the water, so it may contain sharks. Certainly truthful statement in that regard. But if we start to venture towards more of that risk-based approach, we start to think about some of the some of the quantitative aspects that may underpin what the likelihood of that risk occurring may be. So we could think about how often do sharks swim in that shallow part of the water near the beach where we may be at? How long do they stay after appearing in that area? How many sharks could appear at the same time, and so forth. So we can add some of those quantitative elements to really begin to identify and estimate that opportunity for risk occurring. So bringing in that more quantitative evaluation. So as we move forward into the discussion, we'll think about, again, how some of the science can help underpin the risk-based approaches that could be implemented into management practices. So certainly we do know that around the world there can be subtle differences between the different types of priority or major allergens. So there can be certainly regional differences. Certainly many countries have utilized the original Codex list of major food allergens, which involve the priority 8 allergens, including cereals containing gluten. And since that time, again, there may be those regional differences that have developed depending upon exposure scenarios and so forth that may have opportunities for differences in certain areas. So recently, we've in the US have seen an increase in sesame allergy. So as of with our FASTER Act, we had, as of January 1, 2023, the inclusion of sesame as our ninth allergen. Other countries, again, may have those regional differences based on exposure scenarios as well. So again, you might see certain countries have mustard, perhaps celery, lupin, and so forth. So again, important to understand those regional differences, certainly from labeling perspectives as well, if there's export to food products to different countries. We do try to track these differences and provide in a regulatory chart, which you can see the link at the bottom, in which, again, we attempt to illustrate which priority allergens are considered from legislation in various countries. When we think about risk, again, we're thinking about not only those priority allergens and which ones would rise to that inclusion on a priority allergen list, for example, but we're also starting to think about how much is too much if one is exposed. So starting to think about that eliciting dose and that aspect from the clinical perspective, and then how that may translate into operational activities in the food industry, for example, or regulatory activities should reference dose or action level or something akin to that to be implemented. So how much is too much is a key question as we think about all that perspective and what science and clinical evaluations have been put in place over the years. So I like to use this slide from two leading allergists in Europe that we've collaborated over the years that have done food allergen challenge studies, where they've done double-blind placebo-controlled food challenges with that offending allergen. And this example shows for peanuts, where we could see that there's individuals that could react to as little as maybe 0.2 milligrams of peanuts or 0.05 milligrams of peanut protein, considering the 25% protein in peanuts traditionally, all the way up to several grams of consumption that would elicit an allergic response. Food allergies are interesting from a clinical perspective and that dose response effect, as we've seen that all of these individuals may react to different doses individually, but an individual reacting at 0.4 milligrams has a more sensitivity. Their threshold or their value of eliciting dose is much lower, but someone that could react to 400 milligrams, akin to maybe a whole peanut or even a half a peanut for one of those larger Virginia type peanuts, still equally allergic in profile, but those individuals have a little bit of a higher tolerance to peanut before they hit their initial reactive dose. So we can utilize that information across the population to better understand what the dose response looks like in, for example, the peanut allergic population. Now, I use this slide for peanut because it's a nice illustration, but as you'll see, many of the priority allergens have that similar dose response across the population. So again, we could then look at, again, going back to our question, how much is too much? I think we could see that in the more sensitive subset of the population, milligram amounts could cause reactions. Part per million concentrations, again, can be a little bit difficult on that action level dependent upon how much is consumed. So we'll get into that a little bit more later as well. So again, what we look at when we think about that exposure scenario, if I'm an peanut allergic individual, I have an exposure dose that I might be exposed to, which again is a function of how much I've consumed, as well as how much peanut protein may have been present in that portion size I consumed. That together equals that exposure dose. And fundamentally, if I exceed my threshold, my individual eliciting dose with that exposure, I would be predicted to have a reaction. So we can look at that from an individual perspective, but typically what's done on a public health evaluation would be looking at that population basis. So we can take the information from each individual that has gone through a challenge study and begin to combine that and statistically model that dose distribution. And our group, along with our partners in TNO in the Netherlands, have worked hard to assemble a very robust threshold distribution from various priority allergenic sources that you see on this slide. So we have data from about 14 priority allergens that had threshold dose studies that were undertaken from those individuals within that allergic population. So again, what you can see here is that many of the individuals, as I said earlier, the peanuts distribution is this dotted red line. But we see that many of the other priority allergens tend to cluster together. So we see many of the tree nut allergens clustering together. Sesame, which is newer to the list, again, seems to have similar potency as peanut and several of the other allergenic sources. There are some differences where we see, for example, the purple dashed line is soy. That seems to be less potent overall comparatively to other priority allergens. And shrimp allergy seems to have less potency, meaning it takes a higher exposure to elicit those responses in the overall population. Out to the left, we see mustard that, again, based on only 33 individuals, seems to be a little bit more potent. But I urge some caution and interpretation on that one, as I think additional data, if it would were to become available, probably might shift that one a little bit as well. So again, I think this data and the modeling perspective to see how that looks overall in the population is quite valuable to begin to understand how we could utilize this for evaluation of risk and risk assessment. So again, what we can do then fundamentally from a population basis is start to think about that exposure dose, the scenario of how much is consumed and what concentration was present, and then benchmark that by a reference dose, as it's called. So a dose at which we would, in the population, predict that there would be a reaction, or if the exposure dose is less than, we wouldn't predict that reaction to occur. So we'll talk a lot about how one could arrive at a reference dose in the remainder of the discussion here as well. So again, using those dose distributions, we would start to think about, well, what could we look for when thinking about a reference dose? Well, certainly one wouldn't want to select a reference dose based upon, for example, the dose that we would predict 50% of the population to react, kind of that mid-linear point in our dose distribution. That would be a very high percentage of population potentially at risk. And certainly from perspective of individuals that react to lower doses, they would have significantly higher exposure in that case. So often where the discussions have resonated throughout various international forums have been around the ED1, as we call it, or the ED5. And these are ED, eliciting dose, values that would be predicted for 1% of the population to react, so ED1, or the ED5, at which 5% of the population would be predicted to react at that particular dose. That would mean 99 or 95% respectively of those populations would not react to those doses. Also important and centered to the discussion that we looked at during the FAO WHO evaluation and discussion was really looking around what the severity of response at those respective doses could be as well. So that brings us to the expert consultation that originally started in November of 2020 as a virtual expert panel coming together from individuals around the world with expertise in food allergens and allergen risk assessment and so forth. It was an FAO WHO expert consultation that was convened at the request of the Codex Committee on Food Labeling, who is working through a process to update guidance on precautionary allergen labeling and other aspects of allergen management, in addition to some components within the Codex Committee on Food Hygiene. So this turned into a 5-part series that discussed various aspects of allergen risk assessment, from, again, thinking about what are those priority allergens to is it possible to establish threshold levels or reference doses as we arrived at, to implementation of use of those reference doses for decisions around precautionary allergen labeling. And finally, part 4 was kind of the last report to be developed, and that looked at reviewing and establishing a framework for considerations on exemptions of food products that or ingredients that might be derived from priority allergenic sources. So we'll go at a very high level today on some of the basics that we see here throughout the discussions. These are all published now, so I have those links as well in the slides that you can see. So most of our discussion will center around parts 1, 2, and 3. Again, those were the center of establishing what are those priority allergens. Can we then take those and look for information around thresholds or eliciting doses and distribution modeling? in the population, and then apply those into how that could be used for risk assessment and evaluation of use of precautionary allergen labeling. So part of what we looked at in part one was establishing a criteria to assess the priority allergenic status of various food sources. And we look back to some of the work that was conducted in the original FAO consultation that led to the 1999 Codex Priority Allergen List. And at that time, the panelists did review various aspects on prevalence, potency, and severity that were considered part of the criteria that would be ideal for a framework for establishing priority allergenic status for various food sources. But at the time in 1995, there was very limited information, particularly around prevalence and potency. A lot of the participants that were clinical allergists involved had information on severity and what they were seeing as more prevalent allergens in their own clinics, but really we didn't have that population basis across the world. Today, we have much more information, luckily. The science has advanced significantly, both on the clinical side and on statistical modeling and so forth. So that allowed us to hone in on prevalence, potency, and severity as those three key criteria to help establish that and evaluate various allergenic sources. So we weren't only looking at the current list, but we looked at very broadly a number of different sources of food that have been associated with allergic response and looked to see if they met those key criteria. Again, for today, I can't go into all the details, but I do certainly recommend and encourage you to take a look at the report that we have that goes through the evaluation and details the discussions as well and the approaches taken to establish the framework. Again, we looked at prevalence and as one of the key criteria, looking at what data was available for prevalence of various allergenic sources across the world and wherever it was available. We also scrutinized and looked at the different types of prevalence studies that were available, whether they were challenge positive type surveys or random digital dial surveys where they've tried to normalize based on random calls to participants that may have been involved in those surveys. So we looked at those opportunities, looked across the different codex regions as well to establish if we had insufficient data, low data to high prevalence data, very sound data in that regard. Severity, again, looked at which ones seem to have higher reports of anaphylaxis. And in that case, we looked at anaphylaxis somewhat broadly, meaning that we were looking at any involvement of two or more organ systems, but we were also cognizant of looking at, again, reports of more severe type response that had occurred in clinical reports of reactions in the community. Potency, we leveraged the data that was available from the FARP and TNO science and modeling efforts over the years, in addition to looking at new data that was available and looking at that for any potential differences as well. So that together we modeled and utilized that in the framework to come up with our priority allergen list that's been recommended. So to kind of sum up that work, sesame was recommended as a priority allergen as we saw prevalence in a number of different codex regions. We saw potency, as I showed earlier, very similar to many of the priority allergenic sources. And certainly there was, unfortunately, a number of severe reports of severe reactions and unfortunately even fatalities associated with sesame. Others that were recommended to be included in a revised list were cereals containing gluten from that celiac perspective. We then looked at the IgE-mediated allergy perspective, in which crustacea shellfish, eggs, fish, milk, peanuts, and in this case, specific tree nuts that were recommended to be included. And those were almond, cashew, hazelnut, pecan, pistachio, and walnut. We took into consideration thinking about opportunities for cross-reactivity that may occur from a clinical perspective in close botanical relations, such as cashew and pistachio. Oftentimes you'll see clinical reactivity, cross-reactivity in individuals that have cashew allergy because of the high degree of protein identity seen in pistachios. The same was true for pecan and walnuts. Almond was interesting because we didn't have potency data, but certainly had data for some degree of prevalence, but mostly a higher degree of reported anaphylaxis across a number of Codex regions. And so again, that was important consideration in our evaluation. You will note that soy is not in that list of recommended allergens. That in this case didn't meet the criteria of prevalence, potency, severity collectively to be considered in our view for a global list. But it was noted that there could be some regional differences and considerations as well that could be considered by individual risk managers. So again, the list again is now in front of the Codex Committee on Food Labeling and Codex Alimentarios for consideration of a potential revision to that 1999 list. You'll see in this report that we did develop what we considered a watch list from a perspective of severity. And the expert committee that worked in this area noted that there were what appeared to be a number of an increasing amount of reports of severe response to things like pea, lentils, chickpeas, some of those legumes, particularly where we saw protein concentrates or isolates. So having that higher dose of protein potentially could be changing that allergen profile. So we wanted to note that. Others, such as buckwheat in certain regions, seeds such as sunflower, poppy seed, cottonseed, kiwi fruit, were ones that, again, had some degree that stood out from the severity perspective, but lacked some of the other information on prevalence and potency in these cases. The last one is one that we're seeing in many different regions around the world, which is that alpha-gal response in red meats upon being bitten by certain species of ticks that are causing reactivity. So again, that was one that was considered, again, increasing in severity across in international perspective. Moving into part 2, we then looked at these different proposed priority allergenic sources to begin to think about if it was feasible to establish thresholds or reference doses for these different priority allergens based upon those modeling efforts. And again, in this case, we also looked at the severity of response around the ED1, ED5 in particular, to evaluate if there was fundamentally a difference in severity of response that was observed during these challenges. So again, we leveraged the data that we had available from FARP and TNO primarily, but did look to some additional data from clinical studies that we knew were ongoing or being that were just published as well to help support our evaluations. So the potency perspective and the dose distributions were then utilized for considerations around what that appropriate reference dose could be in this case. Severity was one of the critical aspects that was considered during the discussion, and one of our panel members, clinician Paul Turner from the UK, had worked extensively in the area of food challenges and works in understanding those different profiles and severity of response as well in the allergic population. So he had done previously several systematic type reviews, in which he was looking at that opportunity for anaphylaxis that occurred at various doses based on past studies, many of which were associated with peanut, but we looked at all the allergens that were possible as well. So again, what we're looking at, and intuitively what you see from this pyramid, if we think about centering around our ED1 or ED5, again, we're thinking about 99% of the population not having a reaction, or 95% of the population based on that ED1, ED5. So intuitively, we would say, again, they could have no symptoms because they didn't surpass their eliciting dose. Certainly could have very mild kind of subjective type response that wasn't considered clinically as part of a observed response. It was centered upon those objective reactions. So 99 to 95% of the population would fall in this lower part of the pyramid, if you will. The ED1, ED5 would then look at 1 or 5% of the population that could experience an objective type response during an exposure. With that, though, it was noted in the evaluations by Paul Turner and our further evaluations that of those individuals, 95% of those with an objective response would not have any type of anaphylactic type response. So no involvement of multiple organ systems and so forth. And then it was estimated that perhaps 5% of that 95% may have something akin to anaphylaxis, so multi-organ involvement, but the vast majority would be predicted to have a resolve without any type of treatment. Certainly, statistically, one could never rule out the opportunity for more severe type response, severe anaphylaxis, or fatality, but that would be exceptionally low probability in this. In the review of the committee and the work of Paul Turner and others. So again, when we looked at the data from challenge studies specifically, it was noted that there were no severe symptoms, no anaphylaxis type in any doses that fell below the ED5. So they were all very mild or moderate in looking across those different clinical studies that were included in the dose distribution modeling. So with this, the committee proposed the use of the ED5 based on not seeing any fundamental difference between the type of response and severity of response between ED1 and ED5. So with that, a proposed reference dose was established and recommended to Codex Committee on Food Labeling, as you can see on this summarized table. You can see at the top we have for the tree nuts 1 milligram of total protein from the respective tree nut allergens as that reference dose. For egg, milk, peanuts, and sesame, the dose would be 2 milligrams of total protein from those respective allergenic sources, and so on. Subsequent to our recommendation in part 2 in our report, CCFL came back to ask about some of those other more regionally specific allergenic sources, such as celery, soy, since it wasn't on the priority allergen list, tree nuts, mustard, lupin, buckwheat, and oats as well. So with that, the committee went back to look at what data was available for these non-priority allergens to see if there could be a recommendation on a reference dose. And indeed, for celery, celeriac, and soy, there was sufficient data to model the dose distribution, as you saw earlier in some of our work. So a recommended reference dose was included. For oats, there was no oat-specific reference dose, as it was felt that it wasn't appropriate to do so. We didn't have any indication that oat fell into specifically a priority allergen status. Most of the reactivity was associated with potential gluten exposure. Again, primarily, there was a lot of question around some of those studies that whether there was gluten cross contact from wheat, rye, or barley present. So we didn't, from an IgE-mediated perspective, see that was appropriate to establish a reference dose. For others that you see at the bottom in the green columns, there wasn't enough data to model a dose distribution. So in this instance, we looked at the opportunity to establish what we call a value for risk management, kind of a placeholder that would allow us, based upon information, for example, from tree nuts, based upon what we saw conservatively for walnut, pecan, and so forth, again, as a placeholder. These could be further evaluated in the future should more data become available for such evaluations. Mustard, again, we had some data available, but it was deemed that wasn't sufficient for an accurate dose distribution model. So again, it was based on limited value data, so a value for risk management was put in place. So again, these could be used for those risk calculations, those exposure calculations, but again, we wanted to differentiate those between what we've considered sound established reference dose values based on those robust statistical models. So we have a dose, which is again, a milligram amount of total protein from that allergenic source. But often when we're thinking in practice for in the food industry and so forth, we're looking at, well, what action level, what concentration could be of concern for an allergic individual. Again, we're thinking about how we could implement that into based upon analytical methodology and support as well. So we discussed how to translate reference dose into action level. And again, an action level would be that part per million concentration or a milligram of total protein from that allergenic source per kilogram of food as consumed. So again, to calculate that, one can take the reference dose. So for example, 1 milligram of walnut protein in that source and divide by the amount, the kilograms of food that's consumed during an eating occasion. And IgE-mediated response is that acute type response. We look at it from that single eating occasion rather than a extended period of time, because again, it would be that acute response. So we can calculate action levels based upon those two key components. And for illustrative purposes, we looked at a number of different calculations and provided a chart, as you see here, that's even more expansive, going from consumption of around 10 grams all the way up to, I think we went to almost 999 grams of consumption in those tables, just for illustrative purposes. So again, just as a point of discussion, you will see that action level, that concentration that could be present, could vary depending upon how much assumed consumption could take place. So for example, if I have walnut out on the far right of our columns here, we know that the reference dose that's been proposed based on the ED5 is 1 milligram of walnut protein. If, for example, I consume estimated portion size of 10 grams, again, the calculation for an action level would be 100 part per million or 100 milligrams per kilogram that could be present in that particular food product as consumed. Now, of course, intuitively, as you increase that consumption quantity, that action level will decrease, again, to arrive at that same reference dose. So if I go to a consumption quantity of 100 milligrams, I would only have 10 part per million present to still reach that 1 milligram of protein from walnut in this case. So again, the tables help to illustrate some of those examples and calculations to arrive at action levels. So how do we take this information and the proposed reference doses into a risk assessment, risk evaluation for precautionary allergen labeling? And again, Codex Committee on Food Labeling was in a process of developing guidance that on precautionary allergen labeling and working towards a more risk-based approach for decisions around precautionary allergen labeling. So part 3 works to evaluate how to utilize that data, and a lot of discussion around how you can start to bring in that quantitative nature of the risk assessment into labeling decisions and evaluation. So we provide, again, that snapshot of the overall recommendation that I have here. And I think important is we always dive into thinking about, well, how do we use those reference doses in that quantitative evaluation? But I think taking a step back, one always has to remember that to have a sound allergen control program, it's central to having those robust allergen controls in place to start. So it's really those what I consider qualitative aspects on establishing a sound allergen control program from the supply level through to operations and so forth that are the most important process to help mitigate the opportunity for unintended allergen presence, that cross contact. So again, you have to have appropriate quality control, hygiene, risk mitigation practices in place and feel very comfortable around those mitigation strategies and evaluate where improvements could be made. So again, walking through that would then look at, did I make appropriate risk assessment and risk mitigation? If so, is my unintended allergen presence, if possible, greater than or less than a certain action level or that reference dose? So why is this important? Why do we center around precautionary allergen labeling in this discussion? Why did CCFL and Codex have that interest? Well, I think if you look over the years at a number of different studies that have been undertaken on surveys of how allergic individuals and their caregivers evaluate precautionary allergen labeling, it shows and illustrates from around the world that there's a lot of confusion around those different terms and use with, again, many allergic individuals believing that a lot of these precautionary statements such as may contain traces, process of manufacture, facility, and so forth, really are just used for legal liability type protection. Again, what we see in many of these surveys is that individuals then start to look at the terminology and try to evaluate and make their own risk assessment and risk judgment based upon what they believe constitutes that statement. So, for example, a may contain, we see that overall probably more adherence to avoiding a product that says may contain, where we see, again, somewhere between 60 to maybe 90% of the population avoiding that product. They feel that a may contain provides a greater risk of the presence of allergen or potential presence than something like manufactured in a facility, where we see perhaps 30 or 40 to 70% of the population that may actually ignore the statement and consume the product. So again, what we know based on research that's been evaluated over the years is that there's really no difference in the probability of the allergen to be present, more likely in something that says may contain than manufacturing facility. So from that perspective, they have an equal opportunity for the potential presence of that allergen residue. But yet allergic consumers are making that risk judgment based upon the terminology. So there is that degree of risk, certainly degree of confusion that's coming out of that. So much more transparency is needed to provide more risk communication value to use of these. And where there wouldn't be a need, again, having an underpinning benchmark by which there can be decisions on whether to use that statement or not. So certainly what we see is that's kind of current status quo where, PAL isn't necessarily related to the amount of unintended allergen presence. It can be, again, really a voluntary process that individually each company evaluates and has different degrees of how they would assess that risk and apply a PAL statement. So we see a lot of variability and, again, not a lot of transparency on the use of those statements. So that's, again, driving some of that confusion and even that risk-taking behavior. The proposed approach would be to utilize those reference doses as that optimal cutoff to decide upon when PAL should be utilized based upon, again, that unintended allergen presence, UAP, exceeding that reference dose and providing that opportunity and a more harmonized approach to be used by the industry and food business operations. So going back to our illustration before, again, we really need to, again, reiterate the establishment of those sound allergen control and control programs. What this isn't used for is, again, it's not justifiable. PAL isn't justifiable in case I forgot to clean. That would be a deviation from those allergen control programs. In case I use the wrong ingredient, again, risk profile would be much different in that aspect. So those would be deviations and corrective actions that need to be put in place in those instances, not necessarily what we're talking about for use of a risk-based model for decisions around PAL. It's not justifiable to be better safe than sorry. Again, that drives that uncertainty and variability and certainly confusion within the allergic population. Not justifiable just because I don't want to look further. Again, many regulators throughout the world have underscored that there's a need to have good manufacturing practices, allergen controls, cleaning protocols, and programs in place to help mitigate the opportunity for allergen control or allergen cross contact. So having that allergen control program established and monitored and always in that continuous improvement mode is central to this activity. Again, with this, I would comment that it's all currently being discussed in expert committee through CCFL and more recently, a few weeks ago, in the Codex Alimentarios Commission. So there's, again, alignment and more discussions that are going to ensue on the overall recommendations and what may come into those codes of practice for coming out of Codex as well. I do know that they have requested an extension of the process, so they're a request to extend into 2026. So this will be a work in progress, certainly. I do want to point out that some countries are already considering approaches and transitioning towards utilizing the FAO/WHO reference doses. So I'll point out that the Netherlands, as of January 1, implemented a new policy for cross-contact and considerations for precautionary allergen labeling. So they do want to see that risk-based approach premised upon considerations of the FAO/WHO recommended reference doses. And that becomes fully implemented in January 1st, 2026. Belgium is another example where they have a science committee that has provided opinion around also the use of largely the FAO/WHO reference doses. So again, not put into more of a regulatory context like the Netherlands, but certainly in discussion. The Allergen Bureau of Australia, New Zealand, and their VITAL program, which has been using this risk-based approach for many years in part of their calculations and decisions around precautionary allergen labeling, have transitioned to VITAL 4.0, in which those default values in the calculator are based on the ED5 values. So we're seeing some traction already being taken up in various countries and in different programs and risk calculators based upon those ED5 values. Again, I think there's a lot of great resources out there when you start to think about that allergen control program holistically, and then starting to think about how to integrate some of those more quantitative aspects into your overall risk assessment and risk-based approaches for decisions on your allergen control program. So I highlight a couple different resources here. Certainly VITAL has done a lot and has established several different tools that can be utilized to provide this more quantitative risk-based approach for allergen control. Additionally, ILSI Europe had a working group that I was part of as well, where we came up with a guidance on application of allergen quantitative risk assessment. So this one in particular has a holistic look at allergen controls, kind of those qualitative assessment and where quantitative aspects can be implemented as well. Also has some tools such as a Excel-based risk exposure calculator that can be helpful as well. So again, there are opportunities, are resources out there. I would encourage you to take a look at those as you're starting to consider. As we wrap up today, I do want to briefly discuss part 4. Won't go into a lot of detail here, but I think it is noteworthy, again, that part of what the Codex Committee on Food Labeling was looking for in some of their questions was whether it was feasible to provide a list of exempted derivatives from priority allergenic sources. Again, the committee noted that rather than just providing a list, similar to part 1, it's important to make this future-proof. So additional folks in the future, risk managers, countries can utilize these frameworks that have been established rather than just providing certain list of exempted items. So we put together a process, a framework that could be considered as a kind of a decision tree model to go through in consideration for whether you have data and what amount of data may be needed to help guide through an assessment for consideration of an exemption. So again, this would be done in view of collaboratively with a food business operator putting together a dossier, for example, collecting the data to help support that the level of potential protein, maybe it's not present or it's very low, based upon a risk assessment, would have a low likelihood of exposure risk to the allergic population. That's then presented to risk managers, the regulatory authorities, for evaluation and further consideration. So this alone is not a ticket just to get to an exemption. It's a part of the process, that framework for that consideration by regulators. So again, the purpose was to really try to work on that framework to evaluate different approaches on how to derive that the potential exemptions. So what we looked at was putting together an evaluation and decision tree model based upon different considerations for labeling exemptions, and then based upon and tested that model against derivatives that have been previously granted exemptions throughout various countries or regions in the world. So again, not going into a lot of detail here, but you can read into more of the detail in the report. Again, the decision tree model would be really describing and getting a good basis for the ingredient itself, what it's derived from, what are the process control parameters, how consistent is the product produced, and so forth. And then starting to quantify maybe that total protein in that particular ingredient derived from that allergenic source. So things such as a total protein assay might be appropriate in this case. Again, getting that specification, how many, what's the intended use, starting to think about that exposure scenario is an important part on that as well. Okay, then going into, again, that decision tree, if there's protein present, what does that exposure scenario look like? Is it at or below an acceptable exposure? And again, we'll go into what was recommended as a potential for consideration on a derivative in this case. So in this case, yes or no, is it above or below? Should additional evaluation be made? So again, going on to looking at additional analytical evaluation, maybe looking at protein analysis, amino acid analysis, others. Not necessarily always going to clinical evaluation. I think that can be part of the decision process, but not necessarily the first thing that would need to be done in this process. So again, working through those decision points. So overall, some of the analytical considerations, derivatives, certainly intended for to be used as ingredient can vary considerably in protein level and composition. So one needs to be able to evaluate that. Some are highly processed, for example, where such as highly refined oils, where the process is able to reduce or remove that protein fraction. So understanding how to quantify that is important. Considering fit for purpose nature of the methods as well is an important part of the process. Then looking at kind of, again, comparing the framework towards some of the items that were evaluated previously and have exemptions in various regions, things such as glucose syrups, tocopherol, soybean oil, highly refined peanut oil, lecithins, and so forth, were part of the overall evaluation process. What we saw in comparison to kind of those currently exempted ingredients versus what we see with some of the establishment of a potential reference value or that would provide an adequate margin of exposure would be taking the reference dose from that priority allergenic source that I talked about in parts 2 and 3 and dividing by 30. So reference dose divided by 30 seemed to provide that weight of evidence and again meets that criteria that would not require additional clinical studies to establish a safety assessment or safety profile. So again, this one has much more discussion to be ensued by Codex and so forth. So again, another kind of wait and see on how these recommendations are taken up by Codex and brought forward into maybe guidance and so forth, codes of practice. All right, and yep, I think I covered that one as well. So part of what FAO, WHO has since been working on since the closure of the 5-part series is working on kind of summary brochures to help distill some of the dense information in each of the different reports. So I provide the links to these summary brochures. They're very simple 1 or 2 page summaries of the body of work that's in the different reports. Again, highly recommend taking a look at the reports, but these are a nice starting point to look at as well. It can help kind of get a basis for what's the the overall basis of the body of work in that report. So with that, I appreciate your time and certainly look forward to questions, and we'll send it back to Mauricio.
Thank you very much, Joe, for your presentation. Now we are going with the Q&A section of our webinar. What I'm going to do, Joe, is that I'm going to read the questions both in English and Spanish, and then I will translate your answer for the audience. Muchas gracias a todos. Le estaba comentando al doctor Joe que vamos a pasar entonces a la sección de preguntas del webinar. Lo que voy a hacer en cada caso es que voy a decir la pregunta en español y en inglés y luego voy a tratar de traducir la respuesta que el doctor nos va a dar en cada caso. Entonces, para que podamos aprovechar este tiempo, aún tenemos 24 minutos aproximadamente. La primera pregunta que nos hacen de la audiencia es si el maní se puede considerar como un tipo de nuez, que si todas las nueces, como por ejemplo almendras y avellanas, tienen el mismo tipo de alergeno o si es diferente. So, Joe, the first question is, actually it's a combined question. Are peanuts considered among nuts? Do all nuts, let's say hazelnuts, almonds, have the same allergen or is it different?
Yes, great question. Peanuts are not botanically related to traditional tree nuts. Peanuts are a legume, so they would be more closely related to things such as peas, lentils, chickpeas, soybean, lupin, and so forth. We do see some level of similarity in some proteins, of some of the individual proteins in peanuts that we might also see in things such as soybean and other legumes. But again, the degree of identity, the similarity of those proteins is somewhat limited. Legumes are a very broad classification of botanical plants and seeds, so forth. So just because soy and peanut are both legumes doesn't necessarily mean that we see a high probability of a peanut allergic individual being soy allergic as well. There's more diversity in that botanical classification. When we go to tree nuts, again, pretty broad classification, but we can start to see clusters of different families that kind of cluster pretty closely together. So as I mentioned during the talk, cashew and pistachio are very closely related botanically, tend to have a very similar proteins, so the degree of identity or homology tends to be very high. So we would see that opportunity for cross-reactivity. The same could be said for things such as pecan and walnut being in their own separate classification of closely related botanical tree nuts. But again, tree nuts are a very broad group. So if I look at almonds, I don't often see, you could sometimes see, but not as often as one thinks, cross-reactivity from, say, walnut to almond. So if I'm walnut allergic individual, I'm not necessarily allergic to almonds. And the proteins differ in their degree of amino acid identity. So almonds are more closely related to things such as some of the stone fruits, so peach and apricot and so forth. So you'll actually see, not from a clinical perspective perhaps, but from an analytical perspective, methods that are geared for detection of almond proteins. You might see that analytical cross-reactivity if, for example, apple pit seed is present, or apricot, or peach, for example. So there is a large degree of botanical difference there.
Ok, el doctor Joe lo que nos dice es que el maní no es clasificado como un tipo de nuez. Hay que recordar que pertenece al grupo de las leguminosas, entonces va a estar mucho más cercano en términos de relación con productos como las lentejas, los garbanzos, los guisantes, la soya, por ejemplo. Entonces dice que, por ejemplo, algunos de los alergenos, de las proteínas alergénicas presentes en el maní, pueden encontrarse, digamos, similares en estos subtipos de productos, pero el nivel, digamos, de reactividad entre un producto y otro tiende a ser muy baja. En cambio, en el caso de las nueces, dice aquí que también es un grupo muy diverso en cuanto a composición, en cuanto a presencia de ciertos alergenos, pero que en estos casos sí se pueden encontrar como grupos o clusters, así lo define él, de ciertas nueces que sí pueden tener cierta reactividad cruzada. Menciona específicamente el ejemplo del marañón y de los pistachos, que dice que sí tienen alergenos que pueden tener reactividad cruzada. Y entonces él desarrolla su respuesta diciendo que esto puede tener también algún tipo de implicación a nivel de métodos analíticos, de análisis de alergenos, porque también puede haber reactividad cruzada a la hora de la detección. Y él menciona el ejemplo, por ejemplo, de algunos componentes alergénicos en frutas que pueden tener reactividad cruzada en términos analíticos. Bueno, vamos a ver, creo que tenemos más preguntas por acá. Dice aquí, déjenme un momentito. Dice que si la cadena global de alimentos, bueno, la cadena global de alimentos es muy compleja. Pregunta que si se cuantifica el impacto de esta complejidad en el marco de los alergenos. So, Joe, the second question is, the global food chain is very complex. Do you quantify the impact of this complexity in allergens framework?
Another excellent question. I don't know if I can provide a quantitative answer to the question, but agreed, it is a very complex network and a lot of the work that needs to be done and part of the risk assessment framework is looking at, again, having those sound allergen control programs in place. So I think the question may be moving towards, how do we start to quantify risk from our supplier level? And that can be certainly challenging and part of where I think overall the work needs to be done to build the capabilities and build the communication around allergen cross-contact risk, exposure risk. So again, I think a level of discussion around getting suppliers equipped and up to speed on some of the new risk-based approaches can be central to that evaluation. From there, I think, again, we can start to quantify the process in which, again, using these risk-based approaches can be implemented. And again, a combination of that qualitative, quantitative aspect can be put in place. It's not, again, not an easy task, certainly, because I think there's companies that have multitudes of different suppliers and multitudes of ingredients and so forth. So it's a process, complex process. One of the strategies I think that could be certainly incorporated, and again, thinking about the essence of that risk-based approach, is thinking about which ingredients constitute a high proportion of your product. So in that stratification process, you're focusing in on those that would have kind of the most impact in that allergen profile. If you have something that's used at a very small amount as a sub-ingredient to an ingredient that you then use, the risk profile is much lower because the opportunity to have a dose that would be such that could cause a surpass that reference dose would probably be lower. So focusing efforts on those, highest inclusion could be your primary focus to start, and then working towards all of the ingredients could be a good strategy. So certainly, I think we can get there. It's not to say it's without work, but I think there's strategies and practical ways of considering how to start to approach this.
Ok, bueno, el doctor Joe efectivamente confirma, ¿verdad?, que es un escenario complejo, que es una tarea difícil precisamente modelar este tipo de escenarios. De hecho, él en este momento dice que inclusive no es posible dar una respuesta cuantitativa como tal respecto a este tipo de consideración, pero que todo empieza precisamente con el tema de los proveedores, de la persona que da este tipo de ingredientes e insumos a las distintas compañías de alimentos a nivel mundial, porque es importante empezar a desarrollar capacidad de cultura de riesgo y comunicación de riesgo y herramientas en términos de riesgo desde los proveedores de los distintos ingredientes. Dice que esto no es una tarea fácil y no lo será, pero él de hecho propone, ¿verdad?, que una estrategia que puede ayudar es precisamente la definición de cuáles son los ingredientes o grupos de ingredientes más comunes en la formulación de algunos alimentos, con un proceso de estratificación, de manera tal que se pueda determinar cuáles de esos ingredientes comunes tienen alto potencial alergénico y enfocarnos en ese tipo de esfuerzos sobre estos ingredientes en lugar de otros componentes que aparecen en menor cantidad y que tienen una muy baja probabilidad de causar una reacción alérgica. Bueno, aquí tenemos otra pregunta, dice sobre el etiquetado precautorio de alergenos. ¿Cuál es la recomendación para evaluar el rotulado de alergenos de productos que no tienen alergenos, pero que comparte la misma área de otro producto que sí los contiene? Se trata de líneas de fabricación separadas. Okay, Joe, the question is about the precautionary labeling of allergens. So, which is the recommendation to assess the labeling of for allergens in products that do not contain allergens, but they share the same processing area of other products that do contain allergens. But in this case, we are talking about separate processing lines. What is the recommendation in that case?
Yep. Another great question. I think the overall process would be twofold. You're looking at one, first thinking about the dynamic of the allergen profile or potential for unintended allergen presence, not only in the product and process facility that we're talking about, but the supply as well. So did the suppliers, do they have allergens in their facility? If yes, do they have a allergen control program in place? Asking specifics and audit questions about, if yes, how are they managing? Do they have cleaning protocols in place to make sure they're mitigating cross contact if shared equipment is used and so forth? So that's, again, part of those, what I call more the qualitative aspects of allergen control. It's really those sound process of qualifying allergens, thinking about inclusions, receive those so forth. From there, we start to think about also within the facility now, again, it sounds like in this question or scenario, they're on separate lines, but maybe the concern would be, is there opportunity for cross contact to occur through the environment, such as dust, for example, is a question that often comes up in question. Another area I would look at is what's the opportunity for lines that are in somewhat close proximity? Are there suitable barriers or ways to mitigate the opportunity for one product to get launched over into the next line, for example? So there be that potential for cross contact to occur that way. Dust is one that can be evaluated. I think folks we know that dust gets generated, and certainly in a shared facility could have potential for risk, but not necessarily in all instances. An idle line where dust is allowed to settle over time and accumulate certainly would have opportunity potentially to have enough that the first product being produced could have enough that could be above the reference dose, for example. But if we're if both lines are functioning and in active use, we need to think about, well, what's the amount of dust in the air and how fast can it settle comparatively to all of those open areas of food product contact and how fast that product is running over the particular line at the time. So if it's not an idle line, typically what we see is that dusting is such that the settlement rate is not sufficient and the amount of dust in the air isn't sufficient when you consider how much product is flowing over at a rapid rate. So that's things that we could help with in some of those calculations and provide some insights and some potential analytical approaches that could support that evaluation as well.
But Joe, but in that case, the labeling as such, so it has to mention that the product was fabricated in a facility that also process other products with the allergen. It should be like that.
Not necessarily. So do follow guidance that regulators or others in various regions have provided, but that's again part of the essence of what's being discussed as the risk-based approach. So thinking about all those scenarios I've just mentioned and thinking about quantifying that opportunity, if it's below the reference dose, then it would not be per the recommendations by FAO, WHO. It wouldn't be recommended to use a precautionary statement because you're not at a risk level that would elevate to a risk to the allergic population. The only caveat there, I kind of mentioned it in a way, is about particulates. So if, for example, you have that opportunity for, let's just say you're producing peanuts next to something that has, tree nuts, for example. If that line producing peanuts flips a peanut over into the tree nut line, you have a discrete particulate, a rather high dose of allergen. It's going to be, the frequency is going to be quite low, but the opportunity to raise above that reference dose is much higher. So in that instance, again, you want to think about risk mitigation opportunities. How can we prevent something from going from one line to the other? If there isn't an opportunity or it's exhausted that you haven't found that opportunity, that's where, again, you may consider, because of the particulate nature and that high dose that could be present, the use of a precautionary statement, such as may contain.
Okay. Muy bien. Bueno, el doctor Joe nos da una respuesta amplia a esta pregunta. Dice que es una pregunta muy importante. Dice que con respecto al etiquetado, pues todo tiene que empezar con una evaluación basada en riesgo, como es el tema de la charla. Dice que todo empieza una vez más con el tema de los proveedores. Cada empresa debe revisar el tema de sus proveedores, preguntándose, por ejemplo, si el ingrediente que ese proveedor da tiene alergenos. Y si los tiene, entonces si ellos tienen un programa propio para el manejo de alergenos y cómo es este tipo de programa, ¿verdad? Qué tan estricto es en términos sobre todo de, por ejemplo, aspectos cualitativos, de limpieza, etiquetado, ese tipo de aspectos. Y una vez, digamos, que se revisa el aspecto de proveedores, entonces se revisa propiamente lo que pasa dentro de la empresa como tal, y aún en el caso de líneas de procesamiento separadas, aquí, digamos, hay que evaluar claramente el tema de la posibilidad de contaminación cruzada entre las distintas líneas, sobre todo a través de aspectos ambientales, donde un factor muy importante es el tema del polvo, que tiende a jugar un rol importante en el transporte de alergenos entre distintas líneas de procesamiento, aunque estén separadas, es muy importante, y esto sobre todo tomando en cuenta, por ejemplo, si ciertos lotes son los primeros en ser procesados en el día, ¿verdad? Que son los que tienen mayor riesgo, porque es cuando el polvo se ha acumulado a lo largo del tiempo, y entonces este evento de contaminación cruzada es mucho mayor. Pero también hay otro tipo de preguntas, por ejemplo, si qué tan cerca está una línea de procesamiento de la otra, si existe algún tipo de barrera de contención para evitar este tipo de contaminación cruzada. y también inclusive aspectos de la velocidad de la línea de procesamiento que puede, digamos, impactar el riesgo. Entonces, yo le agregué a la pregunta, ¿verdad? Yo le pregunté entonces, digamos, qué implicación tenía esto en términos de etiquetado, que entonces, en estos casos, igual había que mencionar en la etiqueta que si había que el producto, digamos, fue procesado en una fábrica donde se procesan otro tipo de alergenos o que podría contener alergenos, y él dice que eso depende, ¿verdad? Que si precisamente en una decisión basada en riesgo está claro, ¿verdad? Que en esta evaluación el riesgo de contaminación cruzada es tan bueno que la dosis de referencia en el producto final es muy baja, entonces puede ser que el etiquetado en este caso no sea necesario, pero siempre y cuando se haya tomado esta decisión con un enfoque basado en riesgo. Él dice que el único problema aquí es el tema de las partículas, del particulado, ¿verdad? Del producto con el alergeno hacia la línea de procesamiento del producto que no lo tiene, porque en estos casos, aunque la frecuencia de contaminación es baja, sí hay más chance de que algún producto pueda tener una dosis encima de la dosis de referencia. Okay, Joe, let's continue with another question. I think we have time maybe for one or two more questions. La pregunta es, ¿cuál es el papel de los médicos en el campo de los alergenos? Ellos son parte de los proyectos de investigación porque ellos son los que están en contacto con los pacientes todo el día. So, the question is, what is the role of physicians, involved in the allergens field? Are they part of the research projects because they are in contact with patients every day?
Yeah. So the original challenge studies that underpin the dose response data, the threshold data, certainly were involved in the data acquisition. Again, we aren't clinicians, so we have to rely upon their expertise and skills and opportunity for good clinical access to clinical resources to develop those challenge studies. Moving forward, though, I think part of the other question here may be, what's the role of physicians, nutritionists, dietitians, and others in helping to communicate the proposed risk assessment approaches? I think, again, become central in the discussions and communication, educational part of the process. They would be evaluating patients and helping give them clinical management opportunities and recommendations. So we need clinicians, dietitians, and others and they've been very involved in the process to, again, understand and more broadly communicate what the risk assessment process means in terms of using a reference dose. What does that mean to individuals that might be in that more sensitive subset versus others that are above that ED1 or ED5, for example? So again, that's the clinical community is central to working through the process of involving and enrolling out a sound risk-based approach to, again, help ensure safety of allergic consumers, but look for opportunities to help open up more food choice for allergic consumers.
Ok. El doctor Baumert nos dice que sí, definitivamente el papel de los médicos y de los profesionales en medicina y en clínica es muy importante. Sobre todo, ellos juegan un papel muy relevante en el desarrollo de los datos originales, por ejemplo, de umbrales de alergenos que causan algún tipo de reacción. Usualmente ese tipo de datos salen de estudios clínicos realizados por médicos y también de algunos de los estudios de reto de alergenos para ver dosis, ¿verdad? Los challenge studies. Claramente ellos están respaldados y desarrollados por médicos en su mayoría. Y él, digamos, hace énfasis, ¿verdad? Que la mayoría o no la mayoría, sino que muchos de los expertos en esta área no son clínicos como tales, no tienen formación médica, y de ahí la importancia de contar siempre con el apoyo de este grupo, pero no solamente de médicos como tal, sino de otros profesionales relacionados, como el caso de nutricionistas, expertos en dietas y demás, porque precisamente el tema de la dosis de referencia se fundamenta mucho en la opinión y en la expertise de este grupo. Jania, estoy un poco confundido con esta última pregunta que me decís. ¿Me escuchas? Sí.
Sí, te escucho.
Ajá.
Ah,
Sí, ¿cómo es esta
sí,
pregunta de?
la pregunta es sobre el etiquetado precautorio de alergenos. ¿Cuál es la recomendación para evaluar el rotulado de alergenos de productos que no tienen alérgenos, pero que comparte la misma área de otro producto que sí los contiene?
Ajá.
Está hablando de líneas de fabricación separadas y específicamente sobre alergenos como la soya o el gluten.
Y bueno, pero esa pregunta ya la respondió el doctor. No sé, digamos,
¿Verdad?
si sí,
Sí,
yo creo, digamos, que eso ya quedó ahí.
claro, ¿verdad?
Entonces,
Ujú.
vos me decís si una pregunta
Eh,
más.
sí, hay solo una más, te la comento. Dice, en su recomendación es obligado el realizar análisis y cuantificaciones específicas siempre para cuantificar el contenido de un alergeno en un alimento, o podría ser válido realizar la determinación teórica del contenido de alergenos en función de la composición de la materia prima. O sea, si se pueden hacer, si es obligatorio realizar análisis in situ, ¿verdad? O si se puede hacer un cálculo teórico.
Okay, the last question, Joe, is basically if in all cases you have to actually quantify the amount of allergens present in a product, if you want to go through this risk-based approach, or if you can in some way do like a theoretical estimation of the counting of the allergen in a specific product.
Yeah, good question. I think certainly a good place to start would be with those theoretical calculations. That again could, for my example earlier on thinking about those ingredients in your product formulation that might constitute the highest inclusion, certainly those are ones that you may want to look to do some analysis to help quantify perhaps. But using the approach where you're looking at those lower inclusions, those theoretical calculations could help guide you initially to say, well, this set we need to continue to have that focus on. These we're going to evaluate. We're going to ask the questions on how the suppliers are managing cross-contact potential, all of the fundamentals of allergen control and that program. But we might not calculate that there would be a high enough level that could be based on our inclusion in a product to arrive at a level of risk. So those theoretical calculations can help kind of guide you a little bit in that overall risk-based approach to your evaluation.
El doctor responde, ¿verdad? Que en definitiva, bueno, que es una pregunta muy buena y que en definitiva los cálculos basados más en la cuestión teórica son muy importantes, sobre todo en el proceso inicial de evaluación, ¿verdad? Sobre todo con lo que hablamos del tema de los proveedores y demás, con respecto al impacto que ellos pueden tener en el procesamiento. Este tipo de cálculos teóricos son muy útiles, sobre todo para tomar estas decisiones al inicio, sobre todo para determinar sobre cuál tipo de ingrediente o alergeno es importante establecer un control más estricto y eventualmente proceder con una cuantificación más de tipo analítico o metodológico. Entonces, en resumidas cuentas, él menciona, ¿verdad?, que en el inicio es importante y que posiblemente pues un enfoque combinado es más útil en este caso. Okay, Joe, I think this was the last question. So thank you very much for your time. And I think there are more questions, Hania. Maybe, I don't know if you can share those
Yes,
questions with you later.
yes, I think so that we will be sending the other questions because we are over the time. So as told you before, thank you very much, Professor Joseph. We really appreciate your commitment with us and for joining us today. Thank you, Professor Mauricio, for this Q&A session, extraordinary session for everybody. And yes, because we have time sending the question for the Professor Joseph. So, well, let's finalize in the, we need, debemos invitar a nuestra última, que será mañana. Se llama así Sabores Saludables, el valor nutricional de nuestras preparaciones. Es una sesión específica donde queremos, la experta Rebeca Castillo nos va a hablar de cómo lograr disminuir el contenido de sal, de grasa, de azúcar en las preparaciones que hacemos en casa, porque son las más importantes cuando estamos hablando de estos nutrientes críticos que definitivamente cuando se consumen de más pueden afectar nuestra salud. Y también pues invitarles al curso que iniciaremos en enero. Es un curso sobre nutrición basado en la evidencia que nos va a brindar la Academia Española, bueno, la de nutrición basada en la evidencia que está ubicada en la Academia Española de Nutrición y Dietética. Si quieren conocer más sobre este curso tan importante para todas las personas en Latinoamérica, pueden revisarlo en nuestra página web. Y como siempre, les invitamos a que nos sigan en nuestras redes sociales para que conozcan más de nuestro programa científico. No se pierdan las actividades del 2025 que van a estar sumamente interesantes, las que hemos planificado durante este año 2024. Les deseamos lo mejor a todos, pues, o felices fiestas. Happy holidays for you, Professor Joseph. Thank you very much again, and see you next year. I hope so.
Thank you. Gracias.
Muchas gracias a usted. Thank you very much, Professor Joseph. Thank you, Professor Mauricio. Muchas gracias,
Hasta luego.
Mauricio. Un
Gracias a
abrazo
usted.
fuerte.
Hasta luego.
Bye bye.
Bye, Joe.
Bye.
Bye, Professor.